Good morning, everyone. Can you guys hear me?
Yes, sir. Good morning.
Yes, sir. Good morning, sir.
Can you see the slides?
Yes, sir.
Yes, sir.
Okay, I'll start the class. Today we are going to discuss about ketone bodies. Have you guys heard about it? Anything?
No, sir.
Okay, so these are the ketone bodies which are shown on the slide: acetone, acetoacetate, also known as acetoacetic acid, beta-hydroxy butyrate. We will see what are these. So basically, from today's lecture, you will know what are the types of ketone bodies. Okay, what is ketone body and what are the types of ketone bodies. Then significance of ketone bodies. Then steps of synthesis and utilization. Basically, synthesis and breakdown of ketone bodies, and the condition which causes ketosis. So, what are ketone bodies? Ketone bodies are basically water soluble transportable form of acetyl unit. It is also formed from acetyl coenzyme A. We will see how it is formed. But the ketone is a misnomer. Okay, like all the ketone bodies are not ketone bodies. Basically, like beta hydroxy butyrate is not a ketone. In like in carbohydrate only you would have read, right? Aldehyde and ketone groups. But there are other ketones in the body which are not ketone bodies actually. So like pyruvate and fructose. Basically, we call ketone bodies which comes from acetyl coenzyme A from one particular pathway. So these three molecules are formed one by one. So that's why these are called ketone body. We will see how it is formed. So from where it is formed basically. So like from adipose tissue, fatty acid will be released by the breakdown of triacylglycerol, right? So this fatty acid will be converted to acetyl unit. Now acetyl unit means in fatty acid oxidation we saw that ultimately fatty acids form acetyl coenzyme A end product, right? So that acetyl coenzyme A in the liver forms ketone bodies. And now where else acetyl coenzyme A goes? Acetyl coenzyme A goes into the citric acid cycle also, right? So when the sufficient quantity of glucose carbohydrate is present in body in circulation, acetyl coenzyme A will go and take part in the citric acid cycle. But in the deficiency of carbohydrate, when fatty acid metabolism is more than carbohydrate metabolism, this acetyl coenzyme A will form ketone bodies to give energy. We'll see everything. So there are some terms which you have to know. What is ketonemia? Anything like whether anemia, azotemia. When emia is added, it means the increased concentration of that particular thing. So increased concentration of ketone bodies in blood, that is known as ketonemia. Okay. Now ketonuria means excess excretion of ketone bodies in urine. I think these are like obvious. And ketosis, this condition of excess ketone bodies formed by ketogenesis, which causes derangement in various organ. Okay, so that the whole process is known as ketosis. The whole syndrome will be known as ketosis. So ketonemia and ketosis. Ketonemia just shows that the increased concentration of ketone bodies is present in blood, but the adverse impact of this ketogenesis, excess ketone bodies on the various organ, is known as ketosis. So what is the significance of ketone bodies? Ketone bodies serve as fuel for extrahepatic tissues. It means when there is deficiency of glucose, or the glucose we take that acts as fuel, then the glucose storage in the form of glycogen that also acts as fuel, right? Then gluconeogenesis, other like molecules form glucose and ultimately that gives energy. When all these things are exhausted, then ketone body is generally formed in the liver, but it acts as fuel for extrahepatic tissue. This ketone body is not used in the liver for fuel purpose, but its formation happen in liver, but it's used outside liver. Liver can synthesize but cannot utilize ketone body. We will see why it cannot utilize, because the enzymes required for synthesis, liver has those enzymes, but liver does not have one particular enzyme which is required for the utilization of ketone body. Brain can utilize but cannot synthesize ketone bodies. Okay, so after glucose, we know that glucose is very important for brain, but in a severe condition where there is too much deficiency of glucose, brain is forced to use ketone bodies. Okay, now we'll see how ketone bodies are formed. Ketogenesis, it takes place in liver. So what is the substrate or precursor molecule? I told acetyl coenzyme A. So acetyl coenzyme A comes from where? Fatty acid oxidation. Okay, and enzymes responsible for ketone body are found in mainly in mitochondria. Okay, so in the mitochondria of liver cells, this ketogenesis actually happens. Now we'll see a stepwise. First, I'll tell you the steps, and then we will see the full flow chart. Okay. In first step, two molecules of acetyl coenzyme A will combine together and form acetoacetyl coenzyme A. So if you remember the fatty acid oxidation, this was the last step in reverse direction. Acetoacetyl coenzyme A, with the help of enzyme thiolase, was forming two molecules of acetyl coenzyme A. So this is the reverse of the last step of fatty acid oxidation. This is catalyzed by thiolase. So two molecules of acetyl coenzyme A forming one molecule of acetoacetyl coenzyme A. Enzyme required is thiolase. Second step. This is important because this enzyme is important. Acetoacetyl coenzyme A combine with acetyl coenzyme A. So in the first step, it was two molecules of acetyl coenzyme A combining forming acetoacetyl coenzyme A. In the second step, acetoacetyl coenzyme A combine with one molecule of acetyl coenzyme A and water and gives hydroxy methyl glutaryl coenzyme A. HMG coenzyme A generally it is called hydroxy methyl glutaryl coenzyme A. HMG coenzyme A. This reaction is catalyzed by HMG coenzyme A synthase. This is very important enzyme. This enzyme is exclusively present in liver mitochondria. That's why ketone body synthesis or ketogenesis happen only in liver, because this enzyme HMG coenzyme A synthase is present only in liver mitochondria. And this enzyme has two isoforms. The mitochondrial isoform is required for the pathway of ketogenesis, and the cytosolic isoform of the enzyme HMG coenzyme A synthase is required for the cholesterol biosynthesis. So like next class we will read about cholesterol biosynthesis. So you will see that there also HMG coenzyme A synthase is required, but there the isoform is found in cytosol, and here the isoform which is found in mitochondria is involved in ketogenesis. In third step, HMG coenzyme A is then cleaved to acetyl coenzyme A and acetoacetate. Okay, so this HMG coenzyme A is getting broken down into acetyl coenzyme A and acetoacetate. Enzyme is HMG coenzyme A lyase. Basically, it is causing the breakdown of HMG coenzyme A. So the enzyme name is HMG coenzyme A lyase. Both these enzymes, HMG coenzyme A synthase and lyase, must be present in mitochondria for ketogenesis to take place. That's why it only happens in liver mitochondria. The other two ketone bodies, so first ketone body is formed, right, acetoacetate, and other two ketone bodies are acetone and beta-hydroxy betarate. Acetone and beta-hydroxy betarate are formed from acetoacetate, and acetoacetate is the primary ketone body. So how acetone is formed from the acetoacetate? Acetone is formed by the decarboxylation with the help of enzyme decarboxylase. So by the decarboxylation of acetoacetate, acetone is formed, but this decarboxylation can happen spontaneously also. So the formation of acetone from acetoacetate can be without the some amount of, it can be without the presence of enzyme also, without the presence of decarboxylase. So spontaneously also acetone forms. So that is very important, because this acetone breath, because acetone will come out in breath, right? So like in type 1 diabetes or sometime in type 2 diabetes also, because of decreased glucose in the circulation, more and more fatty acid breakdown happens and lead to increased production of ketone bodies. So this acetone is can be like, basically it comes out in breath, and this is used as crude method of diagnosing individual with untreated type 1 diabetes. So the order of the acetone can be detected in the breath of a person. So now two ketone bodies are formed, acetoacetate, then second acetone by decarboxylation. Now third is beta-hydroxy betarate. So beta-hydroxy betarate is formed by the reduction of acetoacetate. The enzyme is beta-hydroxy betarate dehydrogenase. So first on top acetoacetate will form. From acetoacetate, acetone will form with the help of decarboxylase enzyme or spontaneously also, and from acetoacetate beta-hydroxy betarate will form with the help of enzyme beta-hydroxy betarate dehydrogenase enzyme. And this beta-hydroxy betarate is the most predominant ketone body present in blood and urine. Okay, so this is a maximum amount. In maximum amount, this ketone body is found, beta-hydroxy betarate. So how much beta hydroxy betarate will be present will depend on the ratio of NADH and NAD plus inside the mitochondria. If NADH is more, the liver will release more beta hydroxy betarate. We'll see the full pathway. It's easy. Acetyl coenzyme A plus acetyl coenzyme A with the presence of thiolase enzyme for acetoacetyl coenzyme A. So if you see in reverse direction, you will remember that this was the last step of fatty acid oxidation. Acetoacetyl coenzyme A was forming two molecules of acetyl coenzyme A. So once acetoacetyl coenzyme A is formed, in second step enzyme is HMG coenzyme A synthase forming HMG coenzyme A. Okay, and then by the help of enzyme HMG coenzyme A lyase, it forms acetyl coenzyme A and acetoacetate. And this acetoacetate forms acetone with the help of enzyme decarboxylase or sometimes spontaneously also. Okay, and this acetoacetate forms beta hydroxy butyrate with the help of enzyme beta hydroxy butyrate dehydrogenase. So it's simple. Most probably pathway will never come for you, but even if it comes, don't write anything extra. You have to just write in words acetyl coenzyme A plus acetyl coenzyme A thiolase forming acetoacetyl coenzyme A. Then just write the enzyme name HMG coenzyme A synthase. Then here you write HMG coenzyme A. Then HMG coenzyme A lyase forming acetoacetate. Then decarboxylase acetone. Here beta hydroxy butyrate dehydrogenase beta hydroxy butyrate. Okay, so now ketone bodies have formed. You saw all three ketone bodies. Now we will see how it is broken down for the release of energy, right? Ultimately, this is an energy giving molecule when there is deficiency of glucose. Ketone bodies are utilized by extrahepatic tissue via a series of cytosolic reactions that are essentially a reversal of ketone body synthesis. So ketone body synthesis was happening in liver mitochondria. Ketone body breakdown or ketolysis happen in extrahepatic tissue in the cytosol. Okay, so see the difference. Thus ketones must be reconverted to acetyl coenzyme A in the mitochondria. Then this ketone body has to be converted to acetyl coenzyme A in the mitochondria. Then only it will release energy. We'll see the step, but here you don't have to know the detail. I'll tell you the name of enzyme. Here it is not written. So this is the reversal basically. This beta hydroxy butyrate, if you see from acetoacetate, beta hydroxy butyrate was forming in the presence of beta hydroxy butyrate dehydrogenase. So this is the reverse process. Beta hydroxy butyrate in the presence of enzyme beta hydroxy butyrate dehydrogenase form acetoacetate. Okay, and in the second step acetoacetate will form acetoacetyl coenzyme A directly, not two step with the help of HMG coenzyme A, the synthase and lyase. Here the enzyme required will be thiophorase. So it is this enzyme, thiophorase enzyme, which is not present in liver. So that's why acetoacetate cannot get converted to acetoacetyl coenzyme A. That's why it will further not convert it to acetyl coenzyme A. That's why in liver cannot utilize ketone bodies for the purpose of energy because of the absence of thiophorase. Here is the thiophorase enzyme. So thiophorase will convert acetoacetate to acetoacetyl coenzyme A, and then acetoacetyl coenzyme A will form acetyl coenzyme A with the help of enzyme thiolase. The liver has acetoacetate available to supply to other organ. Because beta hydroxy butyrate dehydrogenase is present, so liver can form acetoacetate, but ultimately it cannot form acetyl coenzyme, and this acetyl coenzyme is the energy giving molecule. So liver has acetoacetate available to supply to other organ because it lacks the particular coenzyme A transferase, and the name of this coenzyme A transferase is thiophorase. So thiophorase enzyme is absent in liver, and that is the reason that ketone bodies are synthesized in liver but utilized in peripheral tissue. So just know, because of the absence of thiophorase enzyme, ketone body cannot be utilized in liver. So the enzyme thiophorase is present at high level in most tissue except liver. Importantly, why this is important, this is good also actually, we will see. Importantly, very low level of enzyme expression in liver allows the liver to produce ketone body but not utilize them, right? Because ketone body production is required. What will happen that if ketone body is produced also in liver and it keeps getting utilized, then it cannot be utilized by the extrahepatic tissue for energy purpose, right? This ensures that extrahepatic tissues have access to ketone bodies as a fuel source during prolonged fasting and starvation, because of the absence of thiopherase enzyme in liver. Otherwise, in liver itself, all the ketone bodies would have broken down. So, lack of this enzyme in the liver prevents the futile cycle of synthesis and breakdown of acetoacetate. The futile cycle means something is getting synthesized and broken down without any net output, right? So that's why the absence of thiopherase is good for the extrahepatic tissue, because they get ketone bodies as fuel source. So for any pathway we have to say regulation. Also here we will see that three ways this ketogenesis, total ketolysis, basically ketone body synthesis and breakdown is regulated. First is lipolysis in adipose tissue. So ultimately, from where this ketone body will form? Ketone body is forming from acetyl coenzyme. From where acetyl coenzyme is coming? Acetyl coenzyme is coming from fatty acid oxidation. From where fatty acid is coming? Fatty acid is coming from breakdown of triacylglycerol. So everything ultimately depends how much triglyceride is present in adipose tissue, how much of that triglyceride is getting broken down, and how much of those fatty acid are coming into circulation from adipose tissue and going into the liver for ketone body synthesis. Okay, so first regulation step is lipolysis in adipose tissue. Ketosis does not occur unless there is increase in the level of circulating free fatty acid that arises from lipolysis of triacylglycerol in adipose tissue. You understand this from the opposite end. So there are triacylglycerol. When we take carbohydrate or fat, ultimately excess glucose and fat gets stored in the form of triacylglycerol in adipose tissue. So when there is decreased glucose or decreased like synthesis of glucose from the gluconeogenesis, this triacylglycerol in the adipose tissue forms free fatty acid by the process of lipolysis. Okay, so this free fatty acid will basically come into the circulation, and if the level of the free fatty acid in circulation increases, more and more fatty acid will go to the liver, and then that fatty acid will get oxidized and it will form acetyl coenzyme. Now that acetyl coenzyme which is forming in liver will go into the ketone body synthesis pathway also and citric acid synthesis pathway also. But if there are excess of acetyl coenzyme A, the citric acid cycle gets saturated. So excess of acetyl coenzyme A go into the ketone body pathway instead of going into the citric acid cycle. So understand the reverse sequence. If you start from adipose tissue, triacylglycerol, you will be able to understand how ketone body is forming, in which condition. When glucose level fall, lipolysis induced by, how lipolysis will happen? Lipolysis happen by two methods basically: decreased insulin and increased glucagon. I had told in fatty acid oxidation also, right? Then synthesis also. So lipolysis induced by glucagon secretion causes increased hepatic ketogenesis. So lipolysis is happening in adipose tissue, releasing more and more free fatty acid and ultimately leading to increased hepatic ketogenesis due to increased substrate free fatty acid delivered from adipose tissue. If you read these two sentences, you will be able to understand how the lipolysis in adipose tissue is connected with the increased ketogenesis in liver. Conversely, insulin, so this lipolysis induced by glucagon was in the starvation or fasting state when glucose is low. But if conversely insulin, when there is well fed state, you are eating normal quantity of carbohydrate, this insulin inhibits ketogenesis, right? How? Via triggering dephosphorylation and inactivating hormone sensitive lipase. This hormone sensitive lipase is required for a breakdown of triacylglycerol. So if hormone sensitive lipase is inactivated during the well fed state by insulin, then triacylglycerol will not be broken down. So lipolysis will not be happening. So if lipolysis is not happening, free fatty acid is not present. Free fatty acid is not present, liver is not getting free fatty acid, and if when liver does not get free fatty acid, ketone body synthesis does not happen. So what ultimately understood from here, when there is sufficient quantity of carbohydrate intake in food and storage of glycogen in the body, ketone body synthesis generally does not happen, and this is regulated by insulin. But when the glucose level falls in fasting and starvation, the action of glucagon takes upper hand, and that glucagon causes more and more lipolysis of triacylglycerol, leading to formation of ketone bodies. Because this is required, right? If there is low glucose, we need some energy fuel, so that ketone body formation helps in the as the energy fuel for the extrahepatic tissues. Now, second level of regulation is fate of fatty acid. Now, we saw that lipolysis of triacylglycerol leads to formation of excess free fatty acid. But what can happen to this free fatty acid? This free fatty acid can get oxidized to CO2. How? This free fatty acid can get oxidized to CO2 or ketone bodies, right? Can get oxidized to CO2 by forming ketone body and by forming acetyl coenzyme and ultimately going into citric acid cycle, or it can form ketone body by going into the ketogenesis pathway, or this free fatty acid can again combine with glycerol and form triacylglycerol and phospholipids. So how this regulation happen? We have seen in fatty acid oxidation that this is regulated at the step of carnitine acyl transferase, or I have given example of palmitoyl acid, right? So carnitine palmitoyl acyl transferase, CPT1 or CAT1. So if more and more fatty acid is present, so that will go into the oxidative pathway. More and more fatty acid will get converted to acetyl coenzyme by oxidation, and this CPT1 enzyme help in the intake of fatty acid into the mitochondria. But if enough quantity of glucose is present, then fatty acid biosynthesis is promoted, right? So that time malonyl coenzyme will inhibit this CAT1. So then the fatty acid oxidation will reduce, leading to reduced formation of acetyl coenzyme, leading to reduced formation of ketone body. So under these conditions, free fatty acid enters the liver cell in low concentration and nearly all esterified to acylglycerol and transported out of liver in very low density lipoprotein. So this you will see in the lipoprotein part. Third is fate of acetyl coenzyme. The acetyl coenzyme formed in beta oxidation is oxidized in the citric acid cycle. This you know, or it enters the pathway of ketogenesis. I told acetyl coenzyme either can go into the citric acid cycle or into the pathway of ketogenesis. As the level of serum free fatty acid is raised, proportionally more free fatty acids are converted to ketone body and less are oxidized via the citric acid cycle. So when more and more free fatty acid is formed, it will lead to formation of more acetyl coenzyme, but more of the acetyl coenzyme goes into the ketone body pathway, synthesis pathway, rather than going into the citric acid cycle pathway. Why? Because citric acid cycle pathway gets saturated because it needs oxaloacetate, right? If you remember the citric acid cycle, that you will see that the one of the important component of the citric acid cycle is oxaloacetate, but this oxaloacetate actually comes from the carbohydrate metabolism. So if there is low carbohydrate and glucose, oxaloacetate is coming low, so that's why citric acid cycle gets saturated. More and more acetyl coenzyme cannot go into the citric acid cycle, that's why all the leftover acetyl coenzyme goes into the ketone body synthesis. Okay, so you see here triacylglycerol, this is the stored fat, right, in the adipose tissue. Lipolysis is happening. We saw various conditions where lipolysis will be promoted and where lipolysis will be inhibited. It will form free fatty acid. This free fatty acid will go into the blood from the adipose tissue. If there is excess free fatty acid because of lipolysis, that will go into the liver also. Here free fatty acid will form acyl coenzyme. We saw activation of free fatty acid forms acyl coenzyme. Then CPT helps in taking this acyl coenzyme into the mitochondria. So now depending on the condition, whether excess carbohydrate is present, excess malonyl coenzyme is present, or excess acetyl coenzyme is present, and what are the situation about the hormones like insulin or glucagon, either this acyl coenzyme can be esterified to acylglycerol, means triacylglycerol. Again it will get oxidized to esterified to triacylglycerol, or it will undergo beta oxidation. If it undergoes beta oxidation, it will form acetyl coenzyme. Now this acetyl coenzyme can again go into either in the citric acid cycle or into the ketogenesis. If sufficient carbohydrate is present, then that will lead to more formation of oxaloacetate, and that oxaloacetate will help in taking this acetyl coenzyme into the citric acid cycle pathway. If oxaloacetate is low because of the low glucose in the body, this acetyl coenzyme predominantly will go into the ketogenesis, forming ketone bodies. Okay, this is the full regulation in short. So these are the three levels which we saw, regulation at 3 level. Okay, So now we understood what are ketone bodies, how ketone body is synthesized, how ketone body is utilized, and then how it is regulated. Next, biological significance of ketone body. So by now I think you know everything only. Ketone bodies serve as fuel for extrahepatic tissues. This you have understood. Mainly for brain. For brain it is very important. Apart from that, kidney and heart also uses. But for brain, so we know that brain is very metabolically active and it is metabolically privileged. What is the meaning of metabolically privileged? Glucose is the best fuel, right? In the sense, especially for brain. So most of the glucose, whether you intake with the help of like food or the production of glucose from gluconeogenesis or whatever, most of it is taken up by brain. So this is a single organ utilizes 60 to 70% of total body glucose requirement. So that's why it is metabolically privileged that first glucose will be given to the brain, and brain for normal functioning will always require some glucose. Even in glucose deficient state, even if little bit of glucose is present in the body, first it will be utilized by brain. Now suppose there is no glucose at all, almost in a starvation and prolonged fasting condition, or a carbohydrate deficient diet, if you are taking more and more fatty acid, brain cannot use fatty acid. So basically by beta oxidation of fatty acid, it cannot form acetyl coenzyme A and it cannot use that acetyl coenzyme A for the energy purpose. Why? Because this fatty acid cannot cross the blood-brain barrier. So that's why after glucose, what becomes important? These ketone bodies and amino acids from the protein breakdown, they become the important fuel for brain. That's why ketone body is very much important in the glucose deficient state, because in glucose deficient state other tissues can utilize fatty acid by beta oxidation, but brain cannot, because fatty acid does not cross blood-brain barrier. First ketone body is formed in liver, that ketone body goes into the brain and utilized by the brain. Now heart muscle and renal cortex also prefer ketone bodies, okay, over glucose. In contrast, see here what is there written: heart muscle and renal cortex, like kidney and heart muscle, prefer ketone bodies for energy production, but brain has to adapt. Basically brain is forced to use. Brain does not prefer ketone bodies. It still prefer glucose, but it is forced to use acetate during starvation, and in diabetes also. We will see diabetic ketoacidosis clinical condition. In prolonged starvation, 75% of fuel need of the brain are met by ketone bodies. So individuals eating diet extremely high in fat and low in carbohydrate or starving. So if high in fat, it will form more ketone body. If low carbohydrate, that will also lead to formation of more ketone body, or starving or suffering from lack of insulin. If there is severe lack of insulin, especially in type 1 diabetes, why it happens? It is because of that there is some defect in the pancreas leading to decreased production of insulin. So we know that insulin inhibits ketone body formation, but if insulin is not present, more and more ketone bodies will form. So that's why we see in diabetes, extreme diabetic condition, especially in type 1, ketone body formation increases, and that condition is known as diabetic ketoacidosis. Ketonemia, increased concentration of ketone bodies in blood. It is due to increased production of ketone bodies by the liver. The production of ketone bodies occur at a relatively low rate during normal feeding. We have seen this. And under condition of normal physiological state. Normal physiological responses to carbohydrate shortage causes the liver to increase production of ketone bodies from acetyl coenzyme generated from fatty acid oxidation. This is basically summing up. This last sentence is summing up why ketone body is formed in the body. It is formed because the shortage of carbohydrate forces liver to produce ketone bodies from acetyl coenzyme for energy purpose. Now I told causes of ketosis. If normal quantity of ketone body is forming, that's okay. But what are the conditions which leads to excessive production of ketone bodies, which leads to some organ dysfunction? So that organ dysfunction caused by increased ketone bodies in the circulation will be known as ketosis. So there are different causes behind the ketosis. Most important and most commonly seen in clinics is uncontrolled diabetes mellitus. Especially type 1 diabetes mellitus leads to diabetic ketoacidosis. Second is starvation ketosis. Third is chronic alcoholism, that also leads to ketosis. Then von Giarchis disease. If you remember these three, that's good. It's these three you have to remember. One GRK, it is type of glycogen storage disorder. Okay, so glycogen cannot be utilized for energy purpose. If when the glucose is low, normally glycogen gets broken down, right? If glycogen cannot be utilized, then ketone body will be formed from fat. Heavy exercise can also lead to require excessive production of energy, so that will lead to excessive ketone body formation. Low carbohydrate diet, like some people take keto diet, right, for weight loss. So there they take low carbohydrate, high fat. So if low carbohydrate, that high fat will form more ketone bodies. So fat storage will be depleted. Then this you don't remember, it's okay. Or toxemia of pregnancy, if any sepsis happens in pregnancy. Now, non-pathological form of ketosis are found under condition of high fat feeding. If you are taking high fat, so that is not pathological in the body, right? Because you are taking high fat, that's why more and more ketone body is forming. And after severe exercise, post-absorptive state. So if you have, like, basically if you have eaten and exercise, so that time glucose can be utilized. But suppose glucose has been fully utilized, okay, and you are doing exercise after that in almost like empty stomach, or when you had your last meal after a long time, like some hour have fast, then what will happen? If you do severe exercise, the fat will be broken down for energy production. So those are not pathology, those are physiological. So only one condition we will see in little bit in detail, that is diabetic ketoacidosis. So it is a state of inadequate insulin level. So insulin is required for keeping the ketone body production in check. But if the state of inadequate insulin level will result in high blood sugar and accumulation of ketones and organic acid. So both things, if insulin is low, it will be doing high blood sugar also in blood, right? What it does, insulin helps in taking up of the glucose present in circulation inside the cell. So insulin takes glucose and it puts those glucose inside the cell, and that glucose ultimately forms glycogen, and it ultimately gets stored as triacylglycerol also. But if insulin is low, more and more glucose will keep circulating in the blood, but that glucose will not be utilized for energy purpose. Because of the absence of insulin, more and more fatty acid will be broken down into ketone body also. So this is the very vicious cycle. This diabetic ketoacidosis is a potentially life-threatening complication. Why? We'll see. It happens predominantly in type 1 diabetes, but can also appear in uncontrolled type 2 diabetes also. Now, when it is like all the time in type 1 diabetes, it's not happening, right? It happens when there is like some other condition at the same time, some other infection, inflammation happens in the body. That time the requirement of insulin increases, but insulin is already low. So then that time diabetic ketoacidosis is exaggerated basically. It may be due to intercurrent illness, like if you have diabetes, you're not controlled well. At the same time, these infections happen, severe infection, influenza, gastroenteritis, UTI and all, infection happens. Then pregnancy, okay. Then inadequate insulin administration, you're not taking insulin properly, or MI, when the circulation itself is deranged, or a stroke, or use of cocaine. Young patient with recurrent episodes of, we know that type 1 diabetes mainly happen in young patient, right? With recurrent episode of diabetic ketoacidosis may have an underlying eating disorder. Young patient anyway have some sort of eating disorder, right? That can also lead to more breakdown of fat, or maybe use of insufficient insulin. Decay results from relative or absolute insulin deficiency combined with counter-regulatory hormone. Because the counter-regulatory hormone means the hormone which counters the effect of insulin. Already insulin is not there. Because insulin is less, then the action of glucagon and counter-regulatory hormones like cortisol and growth hormone increases, right? This decreased ratio of insulin to glucose promotes gluconeogenesis, glycogenolysis and ketone body formation. So all these things will lead to decay. How? If insulin is low and glucagon is high, more and more gluconeogenesis happen, more and more glucose is forming, leading to high blood sugar. Glycogenolysis is also happening, glycogen stored is breaking down, forming more glucose, leading to high blood sugar. Ketone body formation is happening in liver by the breakdown of fatty acid. As well as increase in substrate delivery from fat and muscle. So more and more free fatty acid comes from fat and muscle to the liver. Ketone body have a low pH. Because these ketone bodies are acidic and it has low pH, therefore it causes metabolic acidosis. This metabolic acidosis is very deleterious and can be life-threatening. Initially, I think I took or someone took a buffer, right? I think I took only. That if metabolic acidosis happen, body will try to buffer those, right? Compensation, we read na, compensation can happen with the help of bicarbonate and other mechanism to compensate for acidosis, such as hyperventilation, to lower the blood carbon dioxide level. So by buffering of this ketone body with the help of bicarbonate or by hyperventilation, body will try to compensate, but it cannot compensate for long, right? So then if it is not able to compensate, this metabolic acidosis will become severe, leading to derangement in the full circulation and the metabolism of the body. So it can be life threatening. This hypoventilation has particular term in diabetic ketoacidosis known as Kussmaul's respiration. This is basically difficulty in breathing. Basically, you will have deep, rapid, but very fast, shallow breath. Okay, you are not able to breathe properly. Kussmaul's respiration. And this ketone body also causes like electrolyte through the diuresis. So here you see, I don't know how much you will understand, but try to see. First, how you diagnose DKA. DKA should have three things. Diabetic ketoacidosis, hyperglycemia, almost 500-600 glucose will be there, milligram per deciliter. Then second thing which is requirement for the diagnosis of diabetic ketoacidosis is ketones, and a ketone body you find in urine. So hyperglycemia, increased glucose level in blood, presence of ketone body in urine. Okay, normally ketone body should not be present at all in urine, but if it is present even in minute quantity, and acetosis. The acidosis you will see by blood gas analysis. You will see in either arterial blood or venous blood if the pH is decreased. So if there is increased glucose, presence of ketone in urine and decreased pH in the body, the condition is known as diabetic ketoacidosis. So what are the vicious cycle of this diabetic ketoacidosis? You try to see here. I'll read first. But if you understand the metabolism of carbohydrate, especially carbohydrate and fat fully, you will be able to understand. So because of the decreased insulin and increased glucagon and other counter-regulatory hormone, increased lipolysis will happen in uncontrolled diabetes, leading to increased breakdown of triacylglycerol, leading to increased fatty acids, okay, in plasma, leading to increased fatty acid shunting to liver. In liver, fatty acid will form increased ketogenesis. This ketone body will be released in ketone urine, and you can detect that. And this, because ketonuria is happening, the body will try to buffer these ketone bodies with the help of bicarbonate. And bicarbonate is the alkali reserve. So the bicarbonate will deplete. Alkali reserve will deplete in the body, leading to acidosis. So because of this ketonuria, acidosis is happening. Now what is happening? Because insulin is low, another pathway you see here, insulin is low, so glucose uptake inside the cell will not happen, leading to hyperglycemia in the blood, right? This hyperglycemia will lead to really increased coming out of glucose in the urine. So ketonuria and glycosuria both. This glycosuria will take up electrolytes also along with it, causing loss of electrolytes. So in urine, electrolytes also will be lost. So ketonuria is happening, ketone body is coming in urine, glycosuria is happening, at the same time loss of electrolyte is happening. This electrolyte loss will cause cellular dehydration, volume depletion and impaired renal function. So body will have dehydration also, ultimately leading to impaired renal function also. And this hyperglycemia is not only because of excess glucose in the circulation, it is exacerbated because of increased formation of free fatty acid. And this free fatty acid can form acetyl coenzyme, ultimately that can form glucose. Some of the free fatty acid can form in the gluconeogenesis pathway also. Okay. Now at the same time, if glucose is not able to get utilized in the cell, as the fatty acid will also break and protein will also break, leading to increased production of amino acid. These amino acid will also, the glucogenic amino acid will also form more glucose, leading to gluconeogenesis and hyperglycemia. And this gluconeogenesis will again exacerbate the hyperglycemia, leading to osmotic diuresis and leading to renal dysfunction. So you can see so much problem is happening in the diabetic ketoacidosis. So it is a very life-threatening, severe condition. Okay. So to summarize, ketone bodies, acetoacetate, beta-hydroxy betarate and acetone are formed in hepatic mitochondria when there is high rate of fatty acid oxidation. Okay. So what are the conditions? When there is low glucose, okay, like in fasting or starvation, or decreased insulin like in type 1 diabetes, this will form more. Okay. The pathway of ketogenesis involves synthesis and breakdown of HMG coenzyme A. So important enzymes are HMG coenzyme A synthase and HMG coenzyme A lyase for the production. But for the breakdown of ketone bodies, important enzyme is thiophorase, which is not present in liver, it is present in extrahepatic tissues. Ketone bodies are important fuel for extrahepatic tissue. Okay. Ketogenesis regulates three crucial steps. First is control of free fatty acid coming from the adipose tissue. Second is activity of CPT1 in liver, which will determine the proportion of fatty acid oxidized rather than esterified. Third is the utilization of acetyl coenzyme A between the pathway of ketogenesis and citric acid cycle. So if question comes what is ketosis, I think you will be able to write. If question comes how it is regulated, then also you can write, because mostly it will come 2 or 3 marks. Then if question what are the conditions in which ketosis happen, 3 important you have to remember. What all? Diabetic ketoacidosis, starvation ketosis, then alcoholic ketosis. Okay, so that's it. Okay, so thank you. So next class we will see about cholesterol metabolism and regulation. Okay, I'll end the presentation.
Thank you, sir. Thank you, sir.
Okay, bye.